inorganic nitrogen. So the gross nitrogen mineralization rate, M, it is the most important flux or most important process in the below ground nitrogen cycle, where ammonium is formed from the organic material. And so there is, of course, in the system also some leaching of these inorganic ammonium and nitrate. There's nitrification from ammonium to nitrate and so on. And we can have a lot of gaseous productions as well, which I will not talk about very much today. So as I said, the gross mineralization rate is the most important process in the soil nitrogen cycle. And this is very much controlled by the soil C/N ratio, because the microbes that are active, they need carbon to do their processing. And if these substrates are not adequately available, then there will be less production of ammonium. It's kind of an excess when there's ammonium release. This is when the system is very rich. And so I can say, for a general understanding among researchers for tropical forests, it's generally said that they are nitrogen-rich ecosystems, that they often have nitrogen cycle in excess. They're saturated in nitrogen. It's an open nitrogen cycle with a lot of leaching of this extra production. And so in the systems, we also have nitrogen fixation. So N2 is fixated by soil microbes, and this also channels nitrogen into the soil, but this can be regulated. So here is now a study from the Congo Basin, so not too far from Rwanda, where the group of Marine here studied lowland forests and montane forests and the nitrogen balance in these tropical forests. And so they especially studied the nitrogen input and output, and they were concerned with also the mycorrhizal associations and the abiotic factors in the environment. But they actually concluded that these forests have a very tight soil nitrogen cycle. And this is when we look down here in the diagram below the trees, these arrows that go from soil organic nitrogen to ammonium. So that's the gross mineralization rate. And the one that goes the other way is the immobilization. This is when microbes take up ammonium once again. And these two processes, they are equally large, both in the lowland forest, M and I, and also in the montane forest, M and I, they are kind of equally large. So they say it's a tight nitrogen cycle in these forests, in opposition to what I said before as a general concept. And so they also have some nitrification happening, but not too much leaching in these limited forests. So when they compared the lowland to the montane, they even found that the montane nitrogen cycle was much faster than the lowland cycle. And they looked on the losses of dissolved organic nitrogen, which was higher in forests with Arbuscula mycorrhiza. And they looked on that the losses of inorganic nitrogen was the abundant main loss in the other forest types. So this is an example of a little bit contradictory investigation of tropical forests. But that was the nitrogen cycle. And now I would like to talk about phosphorus limitation, because the plants, they of course also need phosphorus. It's a very important nutrient as well. And we often come back to this synthesis by Tanner from 1998, where they said as a conclusion that lowland forests are limited by phosphorus and many montane forests are limited by nitrogen. And they conclude this based on the leaf N and P contents, as you can see in these graphics. So you see on the x-axis the altitude, so the height up, and you see that the leaf nitrogen, it becomes less when you go up in the altitude. The phosphorus content does not really significantly have a direction, but because of these change in the leaf nitrogen, the N to P ratio alters as you go up the mountain. So what is among the reasons behind this mechanism when you're going up the mountain? Yes, it has to do with pedology, the formation of the soil. So when a soil starts forming, this is at the 0 point on the x-axis, and then on the, as long as you go to the right, you have older and older soil. So it's the time of soil formation. So when the soil is very newly forming, you have a lot of this primary minerals with a lot of really available phosphates in it, for instance, apatite. And then as time goes, this weathers and turns into, for instance, plant biomass, which then with time becomes organic material with phosphorus. So as time goes, the phosphor availability decreases. It's more and more occluded, more and more bound in the soil. And when you are looking up on the mountaintop, you in principle are looking on younger soils, because up here the mountain has not been weathered so much compared to at the bottom, where the weathering processes have been going on for a longer time. So that is one reason behind why phosphorus availability changes along the altitude. So with looking at geological time and the soil formation, phosphor becomes less available for plants. Phosphor limitation increases with time, and at the same moment, then nitrogen limitation decreases. And this is because plants, and in fact all cells, they have a kind of a specific need for nitrogen and phosphorus. And people have looked on these ratios for a long time. The most famous is Redfield, who decided or came to the conclusion that for all cells, it was a very general rule that they had an N/P ratio of 16. So this is what we call the Redfield ratio. And so also leaves, if you look at them globally, and if you look at especially plant leaves that are well nutritioned, they tip on this N/P ratio at 16 or 15. And it's quite generally accepted that when plant N/P ratio is less than 14, the plant is N-limited. But if the ratio is higher than 15, it is P-limited. So this is all based on what the plant needs. So there is now becoming more and more data available and investigations available about plants and phosphorus. And one of these new papers say that the phosphorus is really the most constraining nutrient for photosynthesis in forests around the world. And they can bounce back on the hypothesis that used to exist that phosphorus is not important for photosynthesis, but now they say it really does regulate photosynthesis, and it can influence rubisco directly. It's, of course, part of the molecule, so logically this is an important nutrient. And models should start to take into account systems where there is a low phosphorus or high N/P ratio. Another study here from Yangambi in DRC looked on these nitrogen and phosphorus cyclings in these forests in that area. And they looked across more forests in Central Africa and determined the atmospheric phosphorus deposition. And by modeling, they have found that this phosphorus deposition has really taken up the cycle, or increased the biomass production a lot, and is responsible for biomass increase. So with their model, they also show that phosphorus has been the main limiting factor for growth in these tropical forests. So now this was phosphorus, of course. The last paper here, it was phosphorus deposition, just dropping down with rain or with ashes. And before I talked about how phosphorus comes from the rocks. And now I'm going to continue a little bit on the same idea set here, with that nutrients can come from the rocks or the minerals in the soil. So there is this very interesting hypothesis of weathering of rocks, that this releases nitrogen as well, if the rock is rich in nitrogen. So this diagram here, this is a very general geological big scale diagram, and you shouldn't maybe not pay too much attention to this, But the lithosphere, this is everything with geology. Clearly, the dinitrogen comes up and into the atmosphere. We have an atmosphere which is dominated by nitrogen. And this dinitrogen can be fixated by plant biomass. And then when it is part of the plant biomass and this dies, this biomass can sediment. And like with time, long geological time, a sedimentation of dead plant matter can be sedimented, and diagenesis means formation of new rocks, like sedimentary rocks. So this input to sedimentary rocks can with time also become a release from sedimentary rocks when they weather. And when they weather, the nitrogen can come out and come back into the biosphere and be taken up by plant biomass. And it's a little bit following the same scheme here as for phosphorus that you see below here, that like if this sedimentary rock is rich in nitrogen, in its early weathering years, can still release a lot of it, but with time, it will again become bounded or fixed into other components. This is also a diagram of this, what happens with the soil or rock minerals. At the top, you have sedimentary organic matter. So sedimentary rocks that contain organic matter. This can be leached out to organic nitrogen that's more free, which can then be mineralized into ammonium. And we can also here at the bottom have ammonium as part of the minerals, which can be released into ammonium, and then it can become nitrate. And yeah, as all this is released, it's available for biological uptake. So it's really well known these days that rocks can release organic nitrogen or release ammonium and nitrate. All right, now this is the end of the background. So just to conclude for tropical forests, what I said so far, generally people say there is an open nitrogen cycle and it's very rich in nitrogen in tropical forests. And then if you look a little bit more detailed, then people would hypothesize that lowland forests are limited by phosphorus and montane forests are limited by nitrogen. Contrasting studies can show that there are tight end cycles in some systems, and studies have also shown that this mycorrhizal status is very determining for the nitrogen cycle, whether it leaches nitrogen out already at the organic forms or in the inorganic forms. And also, many studies actually say they are not fully accounting for the nitrogen balance, so there are missing inputs especially or outputs. And so when we are looking more into the mineralization of organic material, here we should always consider that microbial nutrient mining and mineralization is controlled by the availability of carbon in the resource. And it can in fact be controlled by phosphorus availability. And so this last pin here is to add that rock nitrogen is maybe an important input to soil nitrogen. So summing up the phosphorus concepts, phosphorus depositions we saw can provide a needed nutrient, and several models recently suggest that phosphorus is the limiting factor for photosynthesis and growth in tropical forests. Along this first hypothesis with the plant entropy ratio, it's clear that highland has possibly more available P than lowland, and this is from the geology input. And then, the leaf entropy ratio of 15. Here, lowland forests will be more limited by phosphorus, and montane forests limited by nitrogen. So if we put this together and come from the geological angle, we can draw this hypothesis up that if you are on the x-axis, you see from low to high limitation, and on the y-axis, you see from low altitude to high altitude. You can see that in the low altitude, we have actually a low nitrogen limitation, but a high phosphorus limitation. And if you go to the high altitude, if you go to the high limitation here, you see it's at the low altitude for phosphorus and the high altitude for nitrogen. So this we'll come back to. So now I will start to dig into the measurements that we have been looking at in Nyungwe. And first of all, I was searching for some time for geological maps because I want to learn about this geology and what might be the type of geology or minerals that could contain available nitrogen. And so it took a bachelor student to open up the GIS databases with geological information, and she generated this nice map. Maybe it's not so detailed as it could be, and it's something we could actually look more at. But you see that the geology of Rwanda contains a lot of different granites, quartzites, and sandstones, and then the schists. And these schists and the mica schists, they are in fact old sedimentary rocks. So they have a potential to contain nitrogen. And this nitrogen, it can be trapped between the clay layers. Clay is layers of clay particles, and between them often sits potassium, but the potassium ion has exactly the same size and charge as ammonium ion, and therefore they will swap over time. So any mineral that contains a lot of potassium potentially also contains a lot of ammonium. That's one of the secrets behind this nitrogen in rocks. So they Emma, she looked further into Nyungwe here with the map and found these nice maps for the geology. And one of the aims of her study was then to look if rock nitrogen or rock types that potentially contain nitrogen, if this influences the soil nitrogen content, so the soil above the rock. And we looked up in databases, this GADM database, and also some results from colleagues from regions in Nyungwe. And she plotted all the data points of nitrogen in soil, and the white points are low content, the black points are high content. And yeah, so it was the exercise to see would there be high content in above geology that potentially contain nitrogen, but we could not really find good correlation at this time. And it was a good exercise and something we would like to repeat perhaps now that we can accumulate more nitrogen data and also can visit the locations to really get the right notes about the geology from in situ inspection. Okay, but this was more of a theoretical study. After Emma came Hilla, and she was actually doing a laboratory thesis. So she was quantifying the rock nitrogen input through weathering by analyzing inorganic nitrogen leached from incubated rock samples. And so this was rock samples taken from Nyungwe. It was Tobias, he was traveling in the region doing some work in Congo, and then he could pick up some samples. And you see, Hilla also made a map of the geological underground. Here it is not distinguished between which type of schist, but it is a lot of schists and some granite areas and quartz areas. And this, I would say, granites and quartzes does not really contain nitrogen, but just schists. The schists will contain nitrogen. And these were the samples, and they were sampled from outcrops like this along the road. Of course, it's not good to have such a destruction of the wall, but it made it possible to take some of these samples of the clay stone or the schist. And yeah, they would be like this. And so in the lab, Hilla measured the nutrient content, first with XRF. She detected these nutrients, and you can see there is a lot of potassium, for instance. And there was also a lot of iron. And then the samples were crushed further, and the nitrogen content was analyzed. Yes. And so these two samples were really interesting because they contained a high nitrogen percent. And people who have studied rock nitrogen a lot more have come to the conclusion. Someone is talking.
Ellie, can you please mute? Thank you.
It's okay. I didn't know if it was a question. So they came to the conclusion that if you have nitrogen percent higher than 0.01%, it becomes ecologically relevant. This is at a limit where it's such a high concentration that it becomes ecologically relevant. So we thought it was interesting to continue the experiments, and she did crush these rocks. So she powdered the samples, and then she added rainwater on top that we had collected, and shaking this, and it was also shaken on a shaking table. And then you see here on the graphic, on each time point through these number of days, she extracted a sample from the bottle and analyzed it for ammonium concentration. And the one that has the highest ammonium concentration here, it is these samples NR1 and 2 that you saw had also the high nitrogen. So that came nicely together. And yes, first of all, there was a good correlation between the potassium content and the nitrogen content in the rock powder directly. This is this connection that ammonium can replace potassium. And there was also a good correlation between the total nitrogen content in the rock and the amount of both nitrate and ammonium that was washed out into this rainwater. So the conclusions here from Hiller's study was that we found this correlation between N and K. So for the future, it might even be possible to just go and measure potassium because it's a good proxy for nitrogen content. And the rock nitrogen inputs, especially from this red-brown claystone schist. This was at a level that is relevant for plants in the ecosystem. Maybe it's a resource that has been forgotten about and not discovered enough. And so it was a pioneer study, but we could definitely set up a better study that quantifies this input better, so we can upscale it better to ecosystem level. Okay, now I will show some results that Tobias and the colleagues from Belgium came to by investigating soils in Nyungwe. And Tobias has put this red circle up here on the Nyungwe map. This is where the study was carried out. So it was a gross mineralization study of soil, so of mineral soil. They literally took away the top organic layer and studied only the mineral soil for practical reasons. And as you can see on this diagram, M here is again the gross nitrogen mineralization rate. And also here it's very important rate, and it's much bigger than the immobilization rate. So yeah, it's a rich system, you would conclude. It's a high rate, and there's also then a high nitrification rate because of all this substrate coming in. You can have a high nitrification rate, and they also measured a high nitrate leaching. So they concluded that those soils were rich in nitrogen. It was an open soil nitrogen cycle. The root uptake was also measured from the same plots. They actually put the label in the ground in the field site and then sorted the soils afterwards, so they could measure the real root uptake. And the roots preferred ammonium over nitrate. So, but what still needs to be done after the study is that they only could achieve data at a little bit longer time scale because of the practical logistics in the field. So what is still needed is to look at incubations on a much shorter time scale, like shorter than 1 day, because when you calculate the turnover time for the ammonium pool here, it is 1 day, and that was the shortest time step they had. So we need to look more into this in detail. Also, the nitrate pool turnover was 4 days. And as mentioned, the investigation was not done in the top organic soil. So there's something missing here. And now it comes over into the ongoing projects and future plans. So what I think could be, and what we think could be really good to investigate in Nyungwe is still to compare these rates with plant leaf entropy ratio, to look at possible phosphorus limitation, and then to really investigate the mineralization rate in the organic soil layer and compare it with a deeper, and then work on this altitude gradient to try to test the hypothesis about phosphorus limitation on the altitude, and perhaps there is also a shift in the nitrogen limitation. So, okay, so to go a little bit further now, we talked about that there might be a big influence on the nitrogen cycle from the soil, both from the soil content of organic material, but also perhaps from input of nitrogen from any rock material. And I found this very nice map of the Rwanda soils with the USDA classifications. And there are all these soil classifications, and it is a challenge to find out what it means, but there can be fished out important information from all the classifications. So for instance, this alphisol here, which is really abundant in Nyungwe and also in many other places, this is a high fertility soil. And so that's what it's referred to. It's a high fertility soil, but it could still be deficient in nitrogen and phosphorus, it says here. So that is the question, what's in it? And it is highly rich with aluminum and iron, which are elements that can trap nutrients. So that is the alphisol. And then we have two more abundant soils. It's the inceptisol. Inceptisol is very abundant also in Nyungwe and in other places. This one does not accumulate these irons and clays and aluminums and organic material, which would trap nutrients. Then the other interesting, it's the red one, it's the oxysol. So this is always reddish or yellow in color because of the high concentration of iron and aluminum and so on. So that is a soil that could trap a lot of nutrients. And so the last one here is the histosol and vertisol. And already now I mentioned that these are abundant in Makera. And so the histosol, it is really originating from a lot of organic material, for instance, from peat that's starting to turn into soil, and it can be very moist. However, the vertisol, it can contain a lot of clay here as well, which can form very big cracks when it dries out in the dry season. So those are soils that are quite different, and of course you have a variation along these categories. And we looked in a teaching book from your university, I think it was this 4th International Soil Classification Workshop, which had a soil pit between Rwanda and Huya, between Rwanda and Arboretum, and it's R5, and this one was certainly a ferrasol. So yeah, so we have these soil types. Nyungwe and Sigira regions will have inceptisols. Rwanda and Arboretum, they have ferrasols. And Makera, it would be vertisols or histosols. And now you know a little bit about their properties. So this brings me to start to tell you about what we did in February, because we took some soil samples and had a couple of weeks of field work in February. And just to make it clear, from Arboretum in Huye, we sampled 3 from each species, 3 from Pollucius fulva, and 3 from Entandophragma excelsum, and 3 from Eucalyptus microcorys. And then, in addition, we visited Rwanda Tree plots in Makera, Sigira, and Rubona. And here, in each site, we sampled from 3 control plots and from 3 fertilized plots. So I start to explain the samplings in Arboretum. Here we see the 3 sites from Polysias with red, 3 eucalyptus sites with purple, and the 3 Entandophragma with green. And here are some nice views on the species. And so I searched the internet for this information. And the Polysias and the Entandophragma and Eucalyptus, they are all growing since around 1950. So it's long established old forests. And we know the families of these, and we know very little about their root symbionts. We only know that the eucalyptus likely has arbuscula or ectomycorrhiza, and the others we do not know this very much. They can all grow very tall. Polysias, it's a native tree to Rwanda. It can be 25 to 30 meters high. It has this trunk that can be up to 1 meter in diameter, and it branches out like an umbrella at the top, maybe. And Entandophragma, this is really the giant of the rainforest. It's also native. It's a late successional species, while Polysias is an early successional species. And the tallest individuals of Entandophragma, they are up to 80 meters tall and 2.5 meters in diameter. So that's a really fantastic tree. So of course, eucalyptus species, they are all exotic to Rwanda, but they can still grow very tall, like 40 to 60 meters tall. And what are we expecting from these samplings in Arboretum? I looked into the literature where many of your colleagues and maybe some of you are here. You have studied Arboretum soils, and it was in several publications found that the eucalyptus species, it performs some kind of soil acidification. And when there was also a study of exactly Entandophragma and Polysius, it was found that there is a more extractable carbon and nitrogen under these trees than under other species. So, and another study here with a PhD thesis from Brigitte says that when she studied early successional and late successional species, they differed in their plant leaf NP ratio. So it was a higher NP ratio in the late species and lower NP ratio in the early species. So there are certainly some differences between the trees, how they grow, how they affect the soil. So the hypothesis is that for these 3 species, they are growing on the same soil with the same parent material in Arboretum, but through these 73 years, they have been adding organic material to the soil. So they have flavored the soil chemistry through their own litter chemistry. And they may also have different root symbionts. So basically, we expect differences in the soil organic material and in the effect it has on the soils and on the nitrogen cycle and the soil organic matter decomposition. So that was hypothesis for Arboretum. Now I take you back to the map here, this nice elevation map of Rwanda. And if you don't already know, Sigira and Rubona and Makera, they are placed like this, also on an altitude gradient. And this is now the design of the Rwanda Tree experiment. So I should not really be the person to explain you the Rwanda Tree design. It should be some of you who have worked much more with it for many years, but at least you can see here that the 3 sites, they have differences in altitude. It is different what forest you would expect on these altitudes, montane forest or transitional rainforest or a bushland forest. So the Rwanda Tree project is mainly concerned with tree responses to climate change and causes of tree mortality and it wants to promote tree plantation using native species. And so there is this setup with irrigation and sheltering against rain, and fertilization. So when we visited these sites, as I said, we only looked into controlled soil and fertilized soil because it was simply not possible to sample everything. And so we focused on this factor. And for example, in this site, the Segira site, all these names you see in the grid, this is plants. So it's abbreviation for the plant name. So they are planted very specifically. And in the red squares, we have the control soils that have no treatment. And the purple squares, they have the fertilization treatment. And the blue spots, this is where we took soil samples to investigate the nitrogen cycle and the soil organic matter deposition. And because everything is planted so specifically, we know exactly the 4 nearest species to each sampling point, or also the secondary layer, but then we will have all species included. But we can look on this effect from species as well here, from all the sites. So what would be our hypothesis? So for soil organic matter degradation and nitrogen mineralization and nitrification rates, we expect all these rates to be smaller in fertilized plots compared to controlled plots within the same site. And this comes back to what I said in the introduction, that when there is already a rich situation, the nitrogen is less mined, so the microbes are working less to achieve any minerals from the organic material if it's already available. And the rates, they will then also be higher if you have a site with more soil organic matter compared to less soil organic matter. So if you have more soil organic matter, it's more work to get the nutrients out. And then also for these sites, we need to be aware that this iron chemistry in the soil types can perhaps inhibit microbial turnover or the nutrient mining. And then we hypothesized to see differences under the species. But I cannot really, I can tell which one will be higher or lower, but we could perhaps draw some parallels to results from Arboretum, where we exactly investigate 3 different specific species. So I just want to bring up the healthy soil concept. So soil health, it is the capacity of a soil to function as a living system that can support plants and animals and humans. Another definition, the health of soil arises from favorable interactions of all soil components, the living and the non-living. It belongs together, the microbiota, the plants and the animals. So in fact, all living soil, it has soil organic matter decomposition clearly. And so now, maybe I already said this word priming. So what is this priming of soil organic matter decomposition? Well, it starts with that soil microbes, most of them, they use carbon. They use organic carbon as a source of energy for their biomass and for their living. So you see in this diagram, the microbial biomass is here. It's using the soil organic matter to live with. And of course, it's making respiration. But what is the priming mechanism? So yes, so it starts with that we have soil organic matter here, SOM, which is in the soil. And the bacteria and the fungi, they want to decompose this because in here are also nutrients. So they produce enzymes and then they can decompose soil organic matter, but they need energy and they need this from readily available soil organic matter. It can be from green leaves that just fall down and very easy to decompose. It can be from root exudates. And some of this contains sugars, for instance, glucose. So glucose is a test molecule for this. So it can be exuded from roots as well, and it activates these microbes so they can decompose more of soil organic matter. Maybe they're already decomposing some, but if they get extra labile carbohydrates, they will also degrade extra soil organic matter. And that is the priming effect. So clearly carbon and nitrogen mineralization are connected. There is a strong correlation between carbon and nitrogen transformation in soils. And we have this nitrogen mining hypothesis. It says that increased carbon availability induces some nitrogen limitation in the microbes, which then access nitrogen by degrading soil organic matter. And that is the priming effect. It starts up the next step. So gross mineralization is likely strongly controlled by soil priming. So we can also have negative priming. You will see this in the next graph as well, but it means that the soil organic matter decomposition is not stimulated. It goes the other way. So this can be happening, for instance, if we have highly stabilized carbon, for instance, some soil that has char in it, biochar, for instance. There can also be a negative priming effect happening if there's been nitrogen fertilization, because it's not needed to decompose. It's just the extra labile carbohydrates is just used as a energy resource that's burned off quickly. So this graphic here, it shows you the CO2 production. And if you have just the first bar here, CO2 that's produced from some, it's of course produced by microbes. So all of this is produced by microbial respiration. And this could be from any cup we have with water, as in the experiment, I will tell you in a few seconds. Then we can have addition of glucose, so addition of these labile carbohydrates. So the microbes will then respire some CO2 coming from the glucose, but they will also respire CO2 from the soil organic matter. And if they then take more from this, have a higher production of CO2 from soil organic matter, that is the rhizosphere priming effect. They have started off more decomposition. But this could also be negative. That's in the other example here, the last example. So we can calculate this relative priming, and I shouldn't spend too much time, I can see, on explaining these calculations now. But I can show you how the experiment happened. So in these cups are soils from Arboretum and from these Rwanda Tree plots. And here Etienne is conducting the glucose addition experiment. This glucose is 13C enriched. So we have bought this product, 13C enriched glucose, which help us to distinguish the glucose-derived respiration from soil organic matter respiration. And then this soil is standing here incubated, and then we will measure the respiration from these cups. And these are data from the delta ray, the machine that measures the CO2 respiration, CO2 concentration, and delta 13C in the CO2. That is the green line here. So at each step is actually 1 cup being measured. 1 cup, 1 cup. And then we have all this wonderful data. It was measured on several time points. And there is some data management needed, where we're making a key-link plot here, the delta 13C in the CO2 versus 1 over the concentration. And these different data management enables us to extrapolate to what was the delta CO2, the 13C CO2 at the start of the measurement, and what was the concentration at the start of the measurement. And making some tricks with we have to subtract the air that was already in the room. And then using this mixing model, we can calculate our way back to the fraction of air that came from some decomposition. So we can calculate this relative priming. And now I will show you the first preliminary data from this experiment. So here we are with the soils from Rwanda Tree. We have many days to calculate. I have just picked out 2 days, the day 16 after glucose addition, it's this one, and then the day 28 after glucose addition. And you see clearly here that it is a negative priming. And yes, now it is up to us to discuss why we have a negative priming, but we can see that the soil types are the histosoil, the ferrosol, and then this insectisol. So clearly there is an effect of the soil type and perhaps this iron chemistry in the soil that gives the negative priming, except for the Sigera soil, which is not limited by this iron chemistry. Here are the results from the Arboretum, and you can see we have some differences between the species, 16 days and 28 days. Arboretum, it has this ferrosol as well, which has this iron chemistry, the brown color. And clearly you see that when we are taking a look back on what was already studied by colleagues here on the site, there is a difference between soils with excelsium and fulva on their soil organic matter content. And this may be something behind the differences here in the priming effect, that there is a difference in the soil organic matter content. But the preliminary conclusions from the priming experiment is that we dominantly see negative priming. Perhaps the carbon is tightly bound in the soil due to the iron chemistry. The mining by the microbes of organic material for nutrients is not limited by carbon, at least. It does not speed up the process. And of course, from now on, we need to relate this data to the data from nutrient availability and soil organic matter content and total carbon content and so on, which is also soon available. And we need to align with the gross nitrogen mineralization rates and the nitrification rates, because these incubations were also done in parallel to the priming experiment. So you can see all the soil here, they have this very nice color, which indicates that there are at least some iron in the soil, or at least a lot of organic material in the soil. Okay, this brings me to the last slide here, summary for future studies in Nyungwe soils. Clearly, rock nitrogen maybe plays a role in Nyungwe. We need to explore this better and in a quantitative way and relate it to the gross mineralization rate. And this gross mineralization rate should be investigated in more smaller time steps and also in the organic soil. And at the same time, I want to study the microbial nutrient mining with a priming experiment in those soils, which perhaps have positive priming as they are insectisols or alphisols. Okay, but first we really need to learn from the studies we have already done in Arboretum and the specific studies at Rwanda Tree with the different climatic zones. So just to round it up, from the thesis from Brigitte here, according to these the limits here of 15 or 16, some 11 species were phosphor limited, some were nitrogen limited, 5 species, and 4 species had no limitation in the Nyungwe study that she did. So it could indicate that overall the Nyungwe forest is not limited by nitrogen, but by phosphorus. But this is something we could look closer at if we come into this altitudinal gradient, which maybe shows some different response and some different phosphorus limitation along the altitudes. Okay, so now thank you for your attention, and thanks again to everyone who are involved in these studies. Now I hope you would like to discuss with me.
Yeah, thank you very much. That's a great talk with a lot of details. And I think some people have questions or comments. Please, you are welcome to ask or comment. Question or comments? You can also type the question or comment in the chat room or unmute yourself to speak. Both ways are possible.
I'm happy to answer any questions. You can also contact me on email. That's nice if you would like to discuss, if you have some ideas. And I can send you this the presentation as well, so you can look at it again and think about your questions. They're also welcome to this.
Yes, you are right. And maybe I can put your email in the chat room. Is that fine?
Yeah,
Okay.
absolutely. You're welcome to. I have sent you my presentation on PDF. You're welcome to send that around, or if you have a website, you can upload it there. And of course, much of this is very preliminary, so it will develop a lot. Yeah, that's my email address.
Ruiz, in your studies, do you also study the contribution of specific plant species in mineralization rate in both forests, Nyungwe and Arboretum?
Yeah, we do study for the Arboretum. We also study this mineralization rate, where we have soil that is specific for each species. So there we will get some information for this.
Yeah, and also, do you have some recommendation for some species someone can plant on his land or her land to increase soil fertility? Yeah, I know legumes are recommended because they fix nitrogen,
Yes, yes.
but what about the trees?
Yeah, there are some trees that have the symbionts that make the nitrogen fixation, but it's not something I know too much about. I think other people can answer this question much better than me.
Yeah, maybe. Anyone who have the knowledge about this? Maybe none, but
Maybe. Hello, can you hear me?
yes,
Yes.
we can hear you.
Yes. Okay. Yeah, maybe I can tell a little bit about it. Well, we have over the years made a lot of nutrient analysis of different tree species, both in Nyungwe and in this experimental plot. So yeah, so I would say that one of your colleagues, Aloisi Manishim, she is a specialist in this, so she will actually defend her thesis on nutrient components and nutrient composition of leaves in different species, probably in September. So yeah, there will be a thesis soon coming out on this, so that will give the details. So that there is what I can say is that the variability between species is really large, even though they are growing in the same soil. The accumulation of nutrients differs a lot between different species.
Okay, thank you very much. Waiting to see
Welcome.
results from Aloizima Nishimwe's research.
Yeah.
Reading from the chat room, Nirenganya Petit asked, I wonder if the study has considered multi-species interactions to contribute in soil organic matter. Can you respond to that first?
I'm sorry, I didn't hear the question. I was.
Yeah, he's wondering if the study has considered multi-species
Yes, yes.
actually to contribute in
I mean, this is
soil organic matter.
very interesting, and it is very complex. And like Jan said, from the study you have now, you see there's a big difference between species. And from the Rwanda Tree setup, it is really a multi-species setup. So every soil sample has many species influencing. And we can because we know the species and how long they have been growing there, we can somehow couple this to the rates at some point. But that would be really challenging.
Okay, thank you very much. Enoch Ndaishimiye said some agroforestry trees are used for their contribution in nitrogen fixation. Yes, that's what I can guess as well. But yeah, can you tell us about some examples? Yes, he's saying acacia species. Mm-hmm. Others? Do you know others, Enoch? If you can speak about it, I can also give you the space to speak. Are you around, Enoch? Yeah, maybe he's not ready, but we are 4 minutes beyond the 4 p.m. And I would like to take like 2 more minutes. If anyone has a comment or a question, to ask it or comment before I close.
I saw one species here that maybe is nitrogen fixing, the Dombeya torrida. Can someone confirm that? Dombeya torrida. And also Albizia gomifera. These were suggestions.
Oh, where are you getting the species?
I was making a list from
It's
the
in the
species.
chat.
Yeah, go ahead.
You want to speak, Bev? You are welcome.
No, it's okay. It's okay.
Okay.
Yeah. Well, I can just say that Albizia gumifera, yes, that's a nitrogen fixer, but probably not Dombia. But Dombia rotundifolia do have really high nitrogen levels in
I think that was
the leaves. Yeah,
that's why we thought about it.
yeah.
Okay. Yeah.
But it's also some of these root symbionts. You can sometimes decide that the species has got it, but then you get to places where it does not exist, the symbiosis anyway. So you can never be completely sure. You need to have the right circumstances in the soil for this rhizobium to thrive well. So I don't know if that can be a challenge in agricultural soils of these soil types, if they can really exist in these soil types.
Maybe we need more studies about that, and also I think Aroizima Nishimwe's research will contribute in this body of knowledge. I would like to close the seminar by here if no one else has a question or a comment. Thank you again, Louise, for speaking to us. Thank you, Gothenburg team.
Thank you very much.
Together with us. Thank you all participants. Actually, it is nice to have you all. We meet again next Wednesday. We learn from each other. We grow together. Remember, if you have something you can share with us, please get in touch with me. I can put you on schedule so you also speak to us.